So previously we had discussed that how this membrane of sperm was made unstable. It was made capable enough to undergo acrosomal reaction, and finally it was made capable enough to fertilize the oocyte. So as it underwent the process of capacitation, its permeability for calcium and bicarbonate ion was increased. And now these calcium and bicarbonate, what it can do is that it will increase the motility of the sperm, as well as it will start a special cascade of events leading to acrosomal reaction. So let's zoom in that what is happening here is that due to calcium and bicarbonate, acrosomal reaction will start. What will happen that the membrane, the acrosomal membrane as well as the plasma membrane of sperm will fuse at multiple points, and at these points of fusion the membrane will break open, and you can say special holes will be punched into this membrane, and through these holes the contents of this acrosome means tip sac that is present at the tip, this the contents of this sac will be released. And what are those contents? There are special molecular cutters. These are proteases, special protein breaking enzymes will be released. And here the most important enzyme is hyaluronidase. You know what hyaluronidase will do is that here, if we see this diagram, that these corona radiata cells, these are embedded into a special ground substance called, what is that called? Hyaluronic acid. So these corona radiata cells are embedded into the hyaluronic acid, and hyaluronic acid is acting as a glue. It is acting as a cement that is you can say stabilizing these cells, that are keeping these cells at their particular position. It prevents these cells from separating from each other, just like the bricks embedded within the cement. So if we break the structure of this hyaluronic acid, these cells will disperse. They will randomly separate from each other, they will disperse. So these molecular cutters or hyaluronidase will be released from acrosome, and they will cut the structure of hyaluronic acid. And as it is done, these corona radiata cells, they will disperse away from each other, and this is called spontaneous acrosomal reaction. I will tell you later on why it is called spontaneous acrosomal reaction, because there is another acrosomal reaction that is non-spontaneous. So then what happens that on Zona pellucida there are special species specific sperm receptors are there. What are these? Human species specific sperm receptors are there, and with that these sperms can bind. Now within these dispersed cells, these hypermotile sperm, they can move through them by their hypermotile action, like a drill machine. They can pass through them and they can attach with the receptor. But here is the sad news for these two sperms. Why there is sad news for these two sperms? Because now they have to penetrate, although they have successfully penetrated the, what is this, the corona radiata, but now they also have to penetrate through Zona pellucida in order to meet the prince's oocyte. But although they may bind with these receptors, but here they cannot punch hole in the Zona pellucida, because they have already lost their weapons, they have already lost their strength, they have already lost these cutters to cut through Zona pellucida, right? So now they are sad and they cannot fertilize the oocyte, and they will be eaten up by female tract. Sad story for them. So then there was this opportunistic clever sperm who went on looking at these other sperms who break open the corona radiata, and now it can get the opportunity and it will bind with this receptor, right? And having those all those intact tools with it in the acrosome, intact head, it can then break the hole in zona pellucida by special cutters. And here an important enzyme, the protease that is used is two are important here: the acrosin enzyme and trypsin like enzymes. They will cut open the zona pellucida and now it can bind with oocyte finally. This is called zona pellucida mediated acrosomal reaction. It is called zona pellucida mediated acrosomal reaction because as soon as the sperm binds with zona pellucida at a particular receptor with their particular ligand, the ligand is not clear yet, but the receptor, the zona pellucida receptor, they are basically called ZP3 receptors. By the way, ZP3 is zona pellucida 3 proteins. There are other proteins as well in zona pellucida, and in the order of its discovery, it was numbered as zona pellucida 3 protein. So this zona pellucida 3 protein acts as receptor of sperm on zona pellucida. As soon as, let's look at that diagram, as soon as it binds with this receptors, these receptors causes a change in the acrosome and its plasma membrane of the sperm that leads to acrosomal reaction. This is not spontaneous acrosomal reaction, it is zona pellucida mediated acrosomal reaction. Now there are, of course, you know that there are two types of acrosomal reaction, but there is extreme importance of spontaneous acrosomal reactions as well, and there is importance of other sperms as well who failed to fertilize, but they opened up the way for this one single fertilizing sperm. They paved the way for this sperm, right? So about 100 or so sperms are required to pave up the way, and without those sperms who were not, although they were not able to fertilize the oocyte, but without the help of those sperms, this one single fertilizing sperm would have never been successful in fertilizing. So that is why they say that other sperms, they help that single sperm in fertilizing, right? So the thing is that although single sperm fertilizes, but that cannot fertilize with the help of other sperm. That is why in a condition when there is oligospermia, the sperm count in a male is reduced, although it is not zero, but sperm count is low. In that condition, the less number of sperm, they cannot fertilize because they cannot help in the fertilizing one sperm by that spontaneous acrosomal reaction, right? What I think of this situation is that the previous sperms, they are like the boys who lose their everything in order to get laid. Even they lose their self-respect in order to get laid, and then in the end another boy with a great sense of humor and with all whistles and bells, he end up getting that girl, right? So that is that funny situation, I think sad situation. And finally it is able to fuse its plasma membrane with the membrane of oocyte. Clear? That is that happy moment for this sperm. You know what? It reminds me of something called Swayamvara. It is an ancient Indian practice that in ancient India, when a princess wanted to get married, so all those who were interested in marrying the princess, they were invited at the event and they were asked to show their extraordinary physical and mental capabilities and strengths. For example, in this picture, in this painting, you can see that this prince, he was asked uh, to shoot the picture of this fish into its eye by this arrow, and only a single prince, only a single candidate who is successful in doing so was selected for as a life partner for the princess. And that is exactly what happens here. Millions of sperms are interested in fertilizing the oocyte, but they have to pass the test. First, they are thrown into that acidic river. What is that acidic river? Vagina. Then they have to traverse through that tough trails of female uterine cavity and fallopian tube. Some of them they get on wrong way, and then here they have to show their cutting tools, right? So that is how the competition is here. After this fusion, the sperm's nucleus, it is pushed into the oocyte's cytoplasm. And as you know that it is rich in calcium, so some calcium also seeps inside the what is this, in the oocyte's cytoplasm, and some calcium also comes through endoplasmic reticulum of the oocyte. And what this calcium will do, it will do many important functions here. But along with that, the what is this, mitochondria of sperm, as well as the tail of the flagellum of sperm also come inside, right? And these mitochondria of sperm as well as this tail, it is destroyed. Only the nucleus is needed here. Only the nucleus of sperm is needed here. And here an important point is that as the mitochondria of sperm is destroyed, it means that mitochondria to the baby, mitochondria for the baby, for the zygote, for the embryo is only contributed by female, by mother. Mitochondria is contributed only by mother, and that is why mitochondrial diseases, it is transmitted not from father but only from mother, right? So mitochondria and tail is destroyed, but nucleus is quite needed here, and calcium comes in. Now what this calcium will do is that there are special sacs or granules present in the cortical region, in the periphery of oocyte. Due to this calcium, these cortical granules, they are dumped outside into this space. What is the name of this space? This is the perivitelline space. What is this? This is perivitelline space, and the space is between oocyte's cell membrane and zona pellucida. By the way, why it is called perivitelline space? Peri means near, vitelline means vitelline membrane. It is near vitelline. Where is the vitelline membrane here? What do you think? There is no vitelline membrane here, but there is perivitelline space here. Well, that makes no sense. Actually, the interesting thing is that this zona pellucida in birds and in some other animals, in small animals, not in mammals, not in humans, this zona pellucida is not zona pellucida. There is a similar structure that is analog of zona pellucida in lower animals and birds, and that is called vitelline membrane. So you can say zona pellucida is vitelline membrane in some other animals, and in humans and other animals there is no vitelline membrane, but rather than vitelline membrane we have zona pellucida. So this space, ideally I think in humans, in mammals, they should be called, this space should be called perizona pellucida space. But I don't know why the embryologist named it as perivitelline space. But truly speaking, it should be perizona pellucida space. Anyhow, the contents of these cortical granules, it is released into this perivitelline space, and what it does is that it digests away, it removes these species specific sperm receptors. Now what is the significance of removal of these receptors? It is just like that this princess, she removes the I am single board, right? So any of the sperm which is still attached, which may be still attached with this receptor, it will immediately fall, and no more sperm can come and bind with this and come inside. So what is the benefit of doing this? This is in order to prevent the multiple sperms from fertilizing a single oocyte. That is called polyspermy. So it is a mechanism to block polyspermy. Why polyspermy should be blocked? Because 23 chromosomes are coming from sperm, 23 are coming from ovum. Total is 46 chromosomes. So normally a human should have 46 chromosomes, but if more than one sperm fertilize, then what it will do? That will lead to aneuploidy. Not aneuploidy. What is that called?
Polyploidy.
That will lead to polyploidy. So in order to prevent that polyspermy from happening, these sperm receptors are removed. And do you remember that this secondary oocyte, it has not yet completed metaphase 2. It is stuck in metaphase 2 phase, metaphase of meiosis 2. Now as soon as it binds there is magic touch of that sperm, there is fusion of sperm's membrane. As soon as that happens, this calcium, it will first remove the sperm receptors, and secondly it will cause the activation of this secondary oocyte, that it quickly completes its meiosis 2 after metaphase, anaphase, and then after cytokinesis the cytoplasm is unequally distributed, right? So normally there should be equal distribution of cytoplasm, but here there is unequal distribution of cytoplasm but equal distribution of chromatids. So one of the cell, this is called definitive ovum, it receives almost all the cytoplasm, and other cell, it receives very little cytoplasm, right? And this is called polar body. There was already a polar body from the meiosis 1, and now there is another polar body. If you want to know more about polar body, you can watch that lecture on polar body. These polar bodies, they are later on removed, but there is a way, this is a way to get rid of the extra chromatids that were there. Now the nuclear membrane reforms, and this what happens to this male's nucleus? You know, initially we had discussed during the spermiogenesis, that this nucleus was tightly condensed, tightly packed like a briefcase, in order to help this sperm keep its size small and pass through these tough trails of female tract. Now as it comes inside this room, inside the ovum's cytoplasm, now it has to relax. Now the chromosomes, they relax a bit, the genetic material, it relaxes a bit, and the nucleus swells up. Now why do you think that why it actually swells up? And it also swells up, the female nucleus also swells up. You know what is the reason? Yes, what do you think? The reason is that now it is having single chromatids, right? Single chromatids. Now before it can undergo the cleavage process, I mean mitosis, in upcoming lecture we will discuss that. Before it can undergo cell division, it must copy its genetic material. It must copy its genetic material, and in order to copy the genetic material, the DNA must be decondensed, the DNA must be relaxed. A highly condensed DNA cannot copy itself. Why? Because to copy the genetic material, the enzymes should be acted upon, and these enzymes cannot act on highly packed up substance, right? For example, you pack up your things in a briefcase to transport it from one place to another, but to use those things, those tools, you have to unpack them, right? So they have to use this genetic material. That is why they have to decondense them, and this decondensation followed by swelling of nucleus, it is called formation of pronuclei. So now we have two pronuclei that are formed, male pronuclei and female pronuclei, and within these pronuclei, these chromatids, copy of these chromatids will be made. And now we have 2 copies of 23 chromatids here in the male pronucleus and 2 copies of 23 chromatids in the female nucleus. So total how many chromosomes we have right now? Yes, 46 chromosomes. So as a result of this fertilization process, we got 46 chromosomes, and a normal human cell, somatic cell, should have, each cell should have 46 chromosomes. So number of chromosomes they have got normal right now. What else will happen? That this membrane, this outer coat of sperm, it will be dissolved, and what are these polar bodies? They will also be destroyed. And now we have this cell called zygote. Now here another important point that should be discussed is about embryo's sex determination, that whether this embryo that is formed, whether it will form a male or a female. Well, it depends upon the type of sperm. This oocyte, it always contains X chromosomes. It always contains X chromosome, but sperm, it may either carry X chromosome or it may carry Y chromosome. So sperm may be X or Y, but oocyte is always X. So if sperm is X, this X will combine with X of oocyte to form double X. What is that? Female. And if that is Y, if that is Y sperm, this Y sperm will combine with X oocyte, and oocyte is always X, to form XY, that is male. So that was about sex determination. Finally, zygote will form. And what is zygote? Zygote is a cell which is formed by fusion of male and female genetic material, which is formed by fusion of male and female gametes, right? Which is formed by fusion of sperm and ovum, which is formed by the result of fertilization. So end result of fertilization is formation of zygote. And by the way, I think it is worth discussing here that what is the definition of fertilization. I think by now it would be very easy for you to understand that fertilization is a process during which male and female gametes fuse with each other to ultimately form zygote. So that is the definition of fertilization. Later on, this corona radiata cell, within a day or two, they will be destroyed and cleavage will happen into this zygote to form the embryo. And zygote is single celled embryo. Next lecture we will discuss how cell division occurs in this zygote. Thank you so much for watching this video.